Just thank you so much for joining us this morning. We're really excited. And without further ado, I'm going to turn the floor over to Dr. Anthony Nichols, the Chief of Head and Neck Surgical Oncology at London Health Sciences Center. Anthony, over to you and your team.
Well, good morning, everyone. Can everyone hear me okay? I'm getting a few connectivity notifications. Can everyone hear me okay? Sorry, can everyone hear me? Pam, can you let me know?
Yep.
Yeah, I can hear you, no problem.
Well, great. So I just wanted to say hello to everyone and thank everyone for coming on today. Special thanks to Pam and Justin and the rest of the foundation for coordinating today. And I just want to say hello to the Kemphes, Lila and Arnie, good to see you. Wendy, the Vol family, Tracy, Uli, Kim, and Betty and Harry Ostrander. Thank you so much for joining the call. And I'll just start my presentation. You know, we really want to make this open, and it's really meant to be interactive. And so everyone should be encouraged to, if you want to unmute and ask questions at any point in time, that would be great. So I want to, well, let me just share my screen first. Can you see my screen? Translational Head and Neck Research Program, everyone? Pam, can you see it?
Yep.
Yep.
Yeah, we can see it.
Yep. All
Okay,
good.
great. Okay, great. Thank you. Yeah, so I'm very proud to lead the Translational Head and Neck Research Program that started back in 2009 when I joined London Health Sciences Center and their foundation. And we're just going to talk a little bit about that. So I want to introduce just a portion of our team, but the people that are working in the lab day-to-day these days. And so, of course, these are the days of COVID. So you can see our lab socially distanced picture. We're trying to be, role model. On the left is Imran, and we're very lucky. Imran's from India, born and raised, but then he did his PhD in Poland. And so he is a multilingual individual. And then he's been with us, I think, since 2017 and working on thyroid cancer. And he's brought his whole family here, including his wife and his adorable son. And Mushfiq Sheikh in the middle there, pictured in the middle, he is from Bangladesh, but he did his PhD in Australia and has been and joined us several years ago and is working on head and neck cancer, which we're going to talk about. And my good friend John Barrett is a senior scientist that I co-lead the program with. And so I also like to introduce Peter, and Peter's gonna be talking later today, and he's our absolute superstar MD PhD student. So he's over 7 years, he's getting a PhD and an MD degree to become not just a physician and a scientist. And overall. So we have other people involved in the programs, but those are the people represented on the calls today. And partially masked because they're working in the lab today and taking time out to join us all. So, you know, one of the main things that we study is head and neck cancer related to human papillomavirus. So historically, head and neck cancers were caused mainly by smoking and drinking, heavy drinking. Some cancers we're not sure about, but over the last couple of years, what we've seen is a dramatic rise in cases due to human papillomavirus. So the same virus that caused cervical cancer can also cause head and neck cancer, specifically of the tonsils and back of the tongue. And, you know, people get infected with HPV. 90% of adults have contact with HPV over the course of their life, usually contracted in their teens and 20s, but most people clear the infection. That the DNA from the virus can hang around for decades and cause cancers years later. This is our experience in London, Ontario, where we looked at every tonsillar cancer that we could find dating back to the 1990s. And in the hashed marks are the human papillomavirus related cases, and in the white bar are the HPV negative smoking related cases. And you can see, although there's fluctuations year by year, overall there's a very dramatic rise in this number of cases of human papillomavirus related disease. And we're seeing just more and more patients in clinic every day. And these tend to be young patients that are younger patients on average, slightly younger, like 64 average age versus 69. We see patients as young as in their 30s and as old as in their 90s that have HIV-related disease. They tend to be non-smokers. And so really a different phenotype of patient than we were seeing before. The important thing is that these patients survival curves, and you're going to start at time 0 on the left, and we go out to 5 years or 60 months on the right. And the HIV negative patients, unfortunately, don't do very well, with about half of them dying from their disease. But the HIV positive patients, the survival of 5 years is about 50%. So, you may be aware, of course, in our time of COVID, there's lots of attention towards vaccination, and vaccinations are indeed medicine's greatest success, despite what the anti-vaxxers say. Vaccines are extremely cheap, extremely effective, and overall very safe. So there is an HPV vaccine that they are giving to children at 10 and below. However, the vaccine only prevents infection, but if you had an infection years ago and just have a little bit of DNA or develop an HIV-related cancer, the vaccine has no effect. People are working on a therapeutic vaccine, but that's still away. So what this means is that there's this huge reservoir of people that have little bits of the HPV DNA hanging around that are going to turn into cancers this year, next year, 10 years from now, 20 years from now. And so certainly for the rest of my career, I'm going to be seeing a lot of these patients, unfortunately, before this problem disappears. And that's only if everyone gets vaccinated, which they're not. So one of the questions is, what's the best way to treat these patients? So the area that these tumors are in the tonsil and back of the tongue. And as you can imagine, those are really important areas for speech and swallowing. And some of the treatments can have profound effects on quality of life, such as impairment in swallowing, up to including needing a temporary or permanent feeding tube in your tummy. Surgery can cause facial disfigurements. There can be pain associated with it, change in voice, and the chemotherapy that we give can cause hearing loss. There can be breakdown of the jawbone, can cause blockage in the neck, radiation. And so with these younger, healthy patients that are usually in the prime of their lives, with a very high chance of cure, we're really looking at what is surgery, and one option is radiation. So in the United States, radiation was the mainstay, but then over the last 10 years, development of technology, including laser surgery through the mouth or minimally invasive transoral robotic surgery, have really taken off. The challenge is that we haven't carefully measured one against the other. We were proud that we developed the first, and for quite some time, the only transoral robotic surgery program in Canada. And then we were involved with mentoring numerous other centers across the country. But what we saw was that there was a clinical arm wrestle kind of every week in clinic. So we have our candidate clinic every Friday. Some of you on the call are all too familiar with that multidisciplinary clinic where there's both a surgeon and a radiation oncologist. And it used to be that everyone got radiation and it wasn't even a discussion. But once we developed our transoral robotic program, it became a clinical arm wrestle. Why should we change our existing standard of care? What evidence do we have that surgery is in fact better? And many places around the world had the same struggle. And so, with the working closely with my superstar colleague, Dr. David Palmer, we opened this trial called the ORDER trial way back in 2012, where we randomized people. And with confidentiality aside, some people on this call are well known to the specific trial where we actually randomized people to radiation versus surgery. And it was a very ambitious thing to do because these types of trials where we randomize to one treatment or another, very, very, very different treatments, are historically very hard to recruit to. And the only way we were able to do this successfully is to admit that the robotic surgery was indeed experimental and radiation was our standard of care and only our surgery in the context of trial, which I think is absolutely the right thing to do. And with this, we were able to bring multiple centers on board. So the University of Ottawa, as well as the University of McGill, helped get their program started, as well as McMaster University. And then people got cutting edge training in the United States at Princess Margaret Hospital, as well as UBC. And we were able to carry out this trial. We even opened a site in Adelaide, Australia, making it an international effort. And so last summer, we were proud that we published this trial, which is truly a world's first, and absolutely put our institution, it was already on the map, but even more on the map. And what it showed is that this, in contrast to much poorly done studies in the United States, is that surgery wasn't vastly superior to radiation. In fact, the swallowing outcomes were slightly worse. They weren't terrible. They were still quite good. They were in the very good range, but not quite as good. And there was different risks to surgery versus radiation. And really, I've done a poll of oncologists afterwards, and this has actually changed how many people care for their patients. About 20% of oncologists said that they shifted more towards a primary radiation approach. And so it's actually impacted care worldwide, which is exciting to see. And that's the ultimate goal of research. And really only a small proportion of research accomplishes that goal of changing treatment. And so we've opened a second trial called Order 2. Because these patients did so well in our trial and others, we've actually toned down the dose of radiation in arm 1 of the trial, and we've toned down the radiation after surgery if it's needed if you get surgery first. And so this trial is open, and unlike the first one where you saw it was just a small number of centers, it truly became a Canada-wide study and open even at 2 sites in Australia, not just 1, and as well as 1 site in the United States. But even though these patients do well, it's not everyone, right? So we said 10%, that's part far from 100. And so you'd really like to know, how do we identify more relevant? So currently we see about 500 patients from Southwest Ontario with head and neck cancer each year, and that is rising. We've measured it. It was only about 250 a decade ago, and now it's 500. And so, out of those, if we're 15% of those patients are doing poorly, we're seeing every other week in clinic, we're seeing one of these HPV patients have their disease come back, spread elsewhere in their body. And so ideally you would want to be able to identify those patients so that you could actually treat them more intensely, not less intensely, and also exclude them. And while we could select for patients that are going to do really well, that we can de-intensify, we can use lower doses of radiation, lower doses of chemotherapy, avoid radiation after surgery in these very low risk patients. But you really have to have robust tests to figure that out. So how do we figure out who is poorly? So you can study the patient and the tumor. So you can study the actual tumor sample. You can also look at blood from patients to look at their genetic predisposition to figure these things out. And so we're well on the way to finding some cutting edge findings, but we need your help to go further. This is a picture of an actual HPV-related tonsil cancer in the throat before we did surgery. So what's been a breakthrough over the last decade or so, is technology. And part of it was enabled through the Human Genome Project. So this is Dr. Eric Lander, who actually had the pleasure of meeting several times when I was in Boston. And he was actually a mathematician who was talking with a geneticist one day, sometime in the 90s, about the problem of sequencing the whole human genome so that we can know what every gene is. And he actually took over the lead of this project. And this project cost $5 billion over about 14 years. But we now know kind of every single little code in the genome. And that's enabled us to develop technology to rapidly analyze tumors as well as genetic conditions in ways that we couldn't before. Because there really is, and in order to look at these tumors comprehensively, we have different tools, right? So for some of you, this may, have to drag up maybe bad memories of science class, right? But I'll bore you with some of these details. So DNA is, what we're born with. We inherit from our, 1 copy of each chromosome from our parent. But then our tissue can be changed. It can change by carcinogens such as tobacco and alcohol. It can be changed by viruses. It can cause mutations in some of these genes. And these mutations, when they get read, turned into something called RNA, a very similar molecule that then gets turned into. DNA can also be modified. We call it the epigenome. It can be modified by little marks on it so that certain areas don't get read, and therefore that gene doesn't get turned on, or some areas can be turned on a lot, so they get read anymore. And so we have tools to study DNA, RNA, the actual protein, and the actual epigenome. And each one of these tells a unique story about the tumor. And when we integrate all these data together, we get a very comprehensive overview of how exactly that tumor works at multiple levels. And they're very complementary information to give a 360-degree view of what's actually going on. So, the study that we're working on, and some of the people in this call have already supported, and I also want to specifically mention our Head Neck Above Cancer Walk organizers. Thank you for all your efforts that have funded these and other projects and infrastructure through the years. And with donor money, we've been able to get a head start on this project. And what we specifically want to do is study 50 patients that did poorly. So remember, again, the majority of patients do well, but over the last decade that I've been here, we've been carefully collecting blood samples and biopsies of the tumor before treatment. And then if patients are unfortunate enough to recur and we're able to access the tumor, we can get a biopsy afterwards. And this gives an opportunity to understand how the tumor evolves. We're also going to study 50 patients that did poorly. So this is a little bit of a busy slide, so forgive me, and I'm just going to walk you through it. This is analysis of the baseline tumor. So this would be as someone comes in with a tonsillar cancer, and we take a little snippet of it before treatment. And then for the small number of patients that recurred, we collected the recurrent tumor. And with this technology developed, from the knowledge of the Human Genome Project, we're able to comprehensively look at every single gene. So we have about 20,000 genes. We can look at every single one. A portion of those can have alterations. We call that mutations. And some of those mutations are important. So when we look at the baseline tumor and we look at the percentage of the tumor that has that alteration, and we compare with the recurrent tumor, we can see that there's a large number of these alterations that both of them share. So this is like, 1,000 specific changes at a specific spot in individual genes. And so the chance of this happening by accident, that these two analysis of this tumor shows by accident, is absolutely zero, absolutely zero. I joke, maybe in bad taste, that if they had this technology when OJ was getting tried, it would have been a different outcome. It's that foolproof that this is just not an accident. All these purple dots that show that there's the exact same change in the exact gene at the exact spot in both the baseline tumor. But what's even more interesting is what we see in orange. So these are, remember, this is percentage of the tumor. So there's some mutations that are present almost in none of the baseline sample, but are greatly, greatly enriched. And so what we now know is happening, because we've proved some of it, is that there's some shared mutations that are present at a very low amount in the original tumor, and then you zap it with chemotherapy, you zap it with radiation, and the part that doesn't have these resistance genes dies away. But the part that has the resistance genes is what grows out and flourishes and turns into the recurrent tumor. And so we believe that a significant number of these genes are functional and cause resistance to both the chemotherapy and radiation. And so we have this handsome guy unmasked here, Mushfiq Sheikh, getting right. He's from Bangladesh and did his PhD in Australia, and then has been with us for several years. And we also had a visiting scholar, Dr. Han, who's a surgeon scientist in Korea, who joined us for over a year. And what they did is we first, the simplest things, you first do a screening in a Petri dish. So we can introduce these genes into cells in a Petri dish, and we can zap them with radiation, treat them with chemotherapy, and figure out which genes create resistance. But I hope no one is a mouse lover here, but we use mice regularly because it's really important to show it's much more robust, is that we actually implant tumors in mice, and then we introduce these alterations into the mice, and we treat them with chemotherapy, we treat them with radiation, and we prove changes in the cells. Only a subset of those changes prove to really cause radiation and chemotherapy resistance in the mouse models. So we've done this with several genes that are important. But what we need to do is validate. So to do the highest level data, because you need to show that your results are reproducible. So, we looked at a small number of tumors at our center and we found these important genes. And then when someone else tries to replicate it at another center, they find no association at all. Unfortunately, that's actually the exception research than what actually happens. And so we've initiated a multi-institutional collaboration across the country. You can see all the stars, whether it's UBC, Edmonton, Calgary, Saskatoon, Winnipeg, basically all the cancer centers in Ontario, McGill, Dalhousie, as well as Memorial University in Newfoundland. And we have them all on board collecting these tumors, and we're getting them all together so that we can validate our findings to prove without a shadow of a doubt that these results are reproducible. And this is what you really need to bring things into a clinic and to really make people believe that what is real. So I hate cocking money. I think it's my parents brought me up to for it to be improper, but I think it's important to realize, how much these studies cost, that it's not an abstract thing. And unfortunately, this molecular analysis, this cutting edge molecular analysis, which we have expertise with our collaborators for analyzing, is costly. And so to study these 150 tumors, remember we're studying 50 patients in blood from patients that did well and were cured, and we follow for over 5 years. Then we're studying 50 patients that we have the baseline tumor and the recurrent tumor in blood. And so that costs actually about $500,000 to do that. Then there's these other assays to look at the protein level, the RNA level, and then we talked about those, the epigenome, how the DNA gets altered by those little marks. And then we need personnel to fund it. So, these are the real costs that I wish I could pay my lab more, but kind of standard costs to fund a postdoctoral fellow who's come, from the other side of the planet. My star MD-PhD student, Peter Zeng. We also have a Genonis analysis that works with our collaborator, Paul Boutros, a veterinary technician for these very complicated mouse experiments. And then to coordinate tumor collection at multiple centers, you can see that it comes up to a pretty pricey price tag. So I think it's important to note that there's other things that you're supporting. You're supporting educational opportunities. So this is actually a small subset. I went through yesterday, and something like 35 trainees have come through our lab, whether they're undergraduate students, we have some high school students, medical students, and graduate students, as well as surgical trainees, such as Kripal Patel, who's now a surgeon scientist in Florida, and people that have gone great to Nicholas Cormier, who's a plastic surgeon in Ottawa, Dr. Han, who's a clinician scientist, and people such as Nicole Pinto, who's now working in drug development at a leading drug company, Morgan Black, who's leading clinical trials here at our cancer center. And so this funding is heavily leveraged to provide educational opportunities to a large number of people, which is I'm really, really proud of. It also has gone to, I just want to say a little bit more about our world-leading thyroid cancer research. So there's people on this call, especially you, Lyle and Arnie, thank you for leading the charge, especially with the Woodstock Group and others for funding our global anaplastic thyroid cancer initiative. So one of the things that I've been also working on over the last decade is this very rare form of thyroid cancer that's almost universally fatal, but it's rare. We only see a few cases each year, but it's stunning. People come in with rapidly exploding neck masses, compressing their airway much faster than the regular head and neck cancer happens. And they often have spread to the lungs. There's often nothing that we can do from. And over 10 years, we've collected over 400 of these very rare tumors. Remember, we see 1 or 2 a year from world-leading sites such as Michigan, Yale, Harvard, MD Anderson, the Mayo Clinic, to Chicago, 2 places in Australia, and really Paul Boutros, I should say, he's acted closely, we've created absolutely novel and groundbreaking world first insights into this cancer and found susceptibilities and potential drug therapeutics about how this cancer arises and how we can treat it better. This, and especially for Arnie and Lala who led this, we have this publication ready. We make this joke about the peer review process. So at the highest level, trying to get these very high level papers. It's been about a year and a half, almost 2 years that we've been working on getting it in, and we're getting there. It's under kind of final review. And, but these very high level reviewers can ask for everything, anything, and more experiments. And so, but we're getting very close to getting this paper accepted, which is going to go into the highest impact of medical journals. And we're really proud of that. And so this is another project with donor funding, which allowed us to generate preliminary data. And again, the grants is a serial application process. Every half year we put them in until we have enough data that you convince them that your project is exciting and has merits, and then you finally get it funded. So that's a major problem, a major point to make, is that donor funds are leveraged, right? So you might look at this, and I went back and took a preliminary look. I should actually, Pam, we should actually find exact numbers, but I believe that our total donations to the Head and Neck Cancer Program over the 11 years I've been here is about $1.5 million. But the amazing thing about that is it's leveraged significant funding. It's let us get, between the other head and neck surgeons and myself, about $10 million of external grants from the Canadian Institutes of Health and industry funding from drug companies, biotechnology companies, and other sources to get us going. And so your money is any donations that are raised by our Head and Neck Cancer Walk are very generous donors, sincerely appreciated. And in addition to the cutting edge work, it actually brings in more money to let us do what we love and try to help people. So in our Head and Neck Cancer team, and those dollars don't just go to me, we have an all-star team. I want to highlight some of our research team, and on the very right is Dr. John Yu, who's a head and neck surgeon, was chief of our department for 11 years, but he's actually now the dean of the medical school, which we're very proud of. He's only the third surgeon in Canada to be the dean of a major medical school. Surgeons are usually too busy running around, but his tremendous leadership of our department, including building our research program, has allowed him to excel, taking over being dean in the middle of a pandemic. And in addition to that, does lots of research, particularly for patient rehabilitation. Our current chair chief is Dr. Kevin Fung, who's doing a equally stellar job running our department. He's an endless source of energy. He is a world expert in education. He started a large variety of educational initiatives that really appeal to this generation of learners, including online modules and interactive teaching platforms. He runs a boot camp where basically every ear, nose, and throat resident from across the country and many from the United States come to receive how to deal with emergencies early in their training, as well as a lot of quality of life research. My colleague, Dr. McNeil, on the left here, is very involved with survivorship, right? So, we can provide cutting edge treatments, but as many of these people on call know, there can be lifelong consequences from head and neck treatment. And so she's really trying to partner with the community to facilitate how we can work better with family doctors, physiotherapists, alternative health providers to care for it better. And then the handsome guy just in there from the left, I don't mean me, but this even more handsome guy, Dr. Adrian Mendez, is our new recruit who has a MD and a PhD. His father is actually a brain surgeon who trained here with the legendary neurosurgeon, Dr. Charlie Drake. And Adrian's, he was also a clinician scientist, and Adrian's followed in his footsteps, albeit in a different field, ear, nose, and throat. And he's very interested in patient quality of life. And so this, was built by first John Yu and then Kevin Fung. You can see, the trajectory of our research program. So one of the metrics we follow is how many papers we publish. We don't just count how many papers, but also the importance of the papers, that they're really groundbreaking findings. And not only have we gone to as few, back in 2003, when there was just 2 head and neck surgeons, there was 4 publications, and then we're now up to 30. And more importantly, these publications are really high quality and actually changing how people do things and changing the field, which is great. So again, why is this HPV project unfunded? So, my experience, so the grant funding is just as tough as, you remember that picture with the scientists walking with the people with baseball bats and spiked instruments down there. So grants are just as challenging. Again, we've had huge success in getting money, but it can often take a long period of time. So my average time to apply, numbers of time that I apply for these major grants is about 6 or 7, and you can only apply every half year. And so this would be the 4th time that I've applied for this HPV positive disease grant, and the reviewers are extremely picky. It's very subjective, and what's critical is you keep things going. And so, as your lab resources drain down, your tendency may be to become conservative. You're like, oh, well, maybe we shouldn't do that next experiment. Or I have this superstar student that's interested in working with me. Maybe I won't hire them. And what donor funding has systematically allowed me to do is, and our program to do, is consistently be aggressive, right? And it's kind of like business. You need to, spend money to make money. And that's, we want to keep, pursuing when we see a cutting edge opportunity to work with other centers, to collaborate. It allows us to forge ahead. We have a big grant application pending. Could we hear in February that we got it? Maybe. But, odds are it might take a few more applications. And over that next couple of years, we want to, while we have some funds in the bank, we don't want to be conservative. We want to keep forging ahead with this project. And the more data we generate, the better we have of getting the grant and moving forward. As you can see how we've leveraged previous funds heavily. So now I'm going to turn it over to this guy again, Peter Zhang. I'd like to introduce him. He's from Guelph, and he did his undergraduate in health sciences at McMaster, and was absolutely top of his class with a perfect GPA. He led numerous initiatives. He did active volunteerism both here and where his parents, where he was born in China. He's won biotechnology competitions, one of which was at MIT in Boston. And so what Peter's doing is the MD PhD program, where they do 2 years of medical school first, and then 3 years of research to get his PhD, and then 2 more years of medical school before going on to further training. So one of these career students. But even while he's been in medical school for his first 2 years, he's created some absolutely groundbreaking data. And so I will turn it over to Peter now. Peter, are you got yourself unmuted?
Yeah, thanks so much, Anthony. And thank you so much to London Health Sciences Foundation for setting this up, and also for everyone to come out for this fantastic event. I appreciate all of your time. So I'm just gonna talk very briefly about some of the projects that I've been working on in the past year or two. And we just lost the screen share.
Can everyone see it again?
Oh yeah, we're back. We're good. Yeah, thanks so much. So in the past year or two, we've generated some very, very exciting data that I'm going to share with all of you today. So I'm just going to talk briefly about them. So on the left side, what we have is a picture of the head and neck region. And as you can start to see very immediately, you don't need to look at the small labels, but you can see that it is very structurally complex. There are a lot of things going on in this very, very tiny region around your mouth and between your neck. However, even though some of these structures are really, really minuscule, they're really, really important for our quality of life. Unfortunately, with a lot of current treatment, head and neck patients often lose their ability to talk, swallow, or eat. And actually, one of my neighbors in Guelph, who I've known for around 15 years, so he actually had head and neck, HPV positive head and neck cancer about 3 years ago, and he actually got it surgically removed at McMaster, and so he can no longer talk and swallow or eat, and he's been on a feeding tube ever since. And it was just really, really heartbreaking to see him after his surgery. We really want to change that. Next slide, Tony. Yeah, so in ideal world, when a patient walks through the door, what we really want to do is we want to separate them into three risk categories: low risk, medium risk, and high risk. And I kind of think of this like, you know, going to McDonald's. When you go to McDonald's to order something, it really depends on how hungry you are. If I'm not really hungry, you know, maybe I'll get some chicken nuggets. But if I'm really, really hungry, maybe I'll get a double Big Mac. And this is the same for a patient who comes in through the door with a tumor. Not every tumor is the same, and some tumors are just more likely to come back, while other tumors are really, really indolent. They don't tend to come back. So why are we treating all these patients very similarly? What we should do is, you know, for the low risk patients, we decrease their amount of treatment. So they can still have their ability to talk, speak, or swallow. For the medium risk, you know, we do what we are doing right now to kind of balance out both the toxicities that comes from treatment, but also preserve some of their function. But for the high risk patients, you know, these patients who cancer are very likely to come back, we might have to make the hard decision to give them a very strong dose of our treatments. But the current problem is we don't really have a way of splitting these patients into these three categories. So a lot of my research has been trying to answer these questions. How do we split these patients into these three categories? Next slide, Tony. Yeah, so to really study this, what we've done is we've collected about 50 fresh patient tumor samples, and we've used this really cutting edge technology called RNA sequencing. And this is a fantastic technology. So it allows us to measure all the RNA made in the tumor cell at a single time. So that's over 20,000. Back, you know, in 10 years ago or 20 years ago, how someone might study this is, you know, they would measure one RNA at a time. So, you know, just studying one RNA might have taken me an entire day or two to look at. But now with these really cutting edge technology, we can actually look at all 20,000 at a single time. And this really has led to huge advances in cancer research. And next slide, Tony. And so through using this technology, what we've started to appreciate is that head and neck cancer really could be split into three categories. So according to the low, medium, high risk. So on the top, we have these immune-rich category of patients. So these patients are, you know, are very likely to do well. They probably have 90 or 100% that they're going to be cured. And you know, you probably heard a lot about the immune system in the past a couple months due to the global pandemic that we're in. You know, we have these antibodies, immune cells. It's basically they really tackle the bad guys. So, for example, you know, when we get infected by a virus or COVID, our immune cell will make sure that we clear all these viruses. The same can be said for a cancer cell. So, our immune system actually has the ability to clear some of these tumors. And when patients have a lot of these immune cells in the tumor, as a result, you know, these tumors get cleared very readily after treatment. Then we have these monocyte-rich group of patients. These monocytes are kind of like these type of cells that really do the dirty work in your body. And these patients have about a medium percent cure in about 80%. And then we have these high-risk patients. So the immune desert, and I really like this picture. It shows this really well. So, you know, at the bottom, we have these immune cells that's trying to get inside the tumor so they could clear them. But these tumor cells are very, very smart. And to prevent them from getting in, they basically set up this desert or this wall of defense so that the immune cells can't get in. And as a result, the tumor can be cleared. And this is why we think these patients are at very high risk of their cancer coming back. Next slide, Tony. So to split these, when patients walk the door, to split these patients into these three categories, we came up with this really actually innovative but also simple approach. So just by looking at 4 proteins, so ZAP70, WFY4, IRF4, CD3, just looking at the 3 levels of these proteins, and then really plugging the values into this equation that we call the UW4 score. And so it's very simple addition. You plug the levels in into this equation, and then you come up with a score. And then just based on the score, you can split the patients into the 3 categories. And this is really simple, but we found a really effective way of really splitting these patients. And we're actually in the process of trying to commercialize this by applying for a patent. Next slide, Tony. Yeah, and with this score, when we're no longer in an ideal world, real world, when a patient walks through the door, we can test these patients using this UW4 score and then split them into low risk, medium risk, and high risk categories. And then we can tailor the treatment to exactly what the patient should need. And this is really exciting. This is what we call precision medicine or precision oncology, where we are no longer treating every patient the same, but we're really trying to figure out exactly what each patient needs to tailor their treatment. And next slide, Tony. And of course, to make sure our results are reproducible, we've actually started this collaboration and got access to data and patients from a variety of centers, University of Calgary, Johns Hopkins, McGill, and to really ensure our results are reproducible in other centers. And this is really, really exciting. We're in the process of doing this, and we can't wait to find out, what these data are going to look like, hopefully very soon. And then next slide, Tony. And again, I would just like to really acknowledge all the patients. This project wouldn't be possible without the patients who agreed to donate their tumor samples. And it just, like every day, coming to the lab, walking through the regional cancer program, just and seeing the patients who are downstairs just really makes a huge difference and adds a lot of meaning to the work that we're doing. And once again, I'd like to thank everyone for coming out to this event. I appreciate everyone's time.